name is Abbyad from Santa Clara University in the San Francisco Bay Area, and I have the pleasure to present our work, SIFT Label-Free Sorting by Droplet Microfluidics. So I work in the field of microfluidics, which is a flow of liquids in small channels, typically 10 to 100 micrometers. We make microfluidic devices. A picture of one of these is shown, and the idea is to do an analysis using less reagents, potentially faster and cheaper. We often talk of the idea of a lab on a chip. So the idea of doing all the operations you might do on the benchtop all within one small disposable device. Now I work in a subfield of microfluidics called Droplet Microfluidics. And we produce little water droplets in oil. In this video, you see the production of these droplets as oil and water are pushed through constriction. Now these water droplets that are from anywhere from picoliter to nanoliter volumes become very good vessels to be able to study chemical reagents or biological reagents such as cells, either in parallel or in series. Now as you see in this video, the droplets flow from left to right. Now, you may be in a situation you want to observe the same droplet or droplet content for an extended period of time. So it would be useful to have ways to control the position and movement of droplets. Now, during my postdoc in Ecole Polytechnique in Paris, a lab I was associated with developed a technique to control the position and movement of droplets. And the idea is that the droplets aren't spherical. They're kind of sandwiched between the bottom and top of the channel, as you see here. Now, if you microfabricate a well, the droplets will expand into that space to reduce their overall surface area. And then the droplets stay confined or positioned in place despite the continued external flow of oil. Now here you see that technology at work. Droplets flow into these wells and get stuck in place. In this particular case, the relative size of the droplets versus the well means that 3 droplets will fit into each well, but this can be adjusted to accommodate for any number of droplets. So in the end, you get this nice petal shape where 3 droplets fit in each well. Now, instead of a well, you can imagine a trench, as you see here. In this particular case, the droplets will follow this trench or this rail like a train on a track. So within this wide microfluidic channel, the droplets will preferentially follow this rail. So between the technologies of these wells or anchors and rails, we can control both the position and movement of individual water droplets. So within the first few years as an independent PI, I used these rail and anchor systems to be able to study individual cellular properties or doing single cell analysis. And this shows you one of the type of assays we would run. So what you have here is you have individual water droplets in an array. So the droplets are about the same size as the wells over here, and individual cells are circled in red. Now afterwards, we can run a time series. And in this particular video, we've added a reagent where lactate produced from the cell gets secreted and turned into a fluorescent product. So here you see a time series, and the droplets that contain cells get brighter from fluorescence. And what we can do here is then we can back from the fluorescent signal over time, we can calculate how much lactate was released from individual cells. So this is a neat technique because it allows us to do single cell analysis in a nice, easy motif. But it's kind of a dead-end measurement. In what sense? It's difficult then to do the next experiment or analyze why these individual cells are showing different metabolic rates. So what we really want to do was develop a way where we could actually sort cells based on one of their properties. Now, the whole story I'm going to talk to you about today was kind of discovered by accident. We were studying different droplet properties, and we came across this particular phenomenon. What you're going to see here is first droplets at high pH, so pH 7.35, are going to flow into this system and into these wells. But what happens afterwards, they're going to flow in droplets at a lower pH. And what they're going to do is they're going to bump out all the droplets at the higher pH and get stuck into place. So once these lower pH droplets come in the system, they get stuck. So we want to study what was happening here. So we did the following experiment. We measured the interfacial tension, or if you want surface tension, as a function of pH for one particular chemical system. And we did this by a technique called pendant droplet, where we use a droplet shape to determine the interfacial tension. And what we observed was that as the pH decreased, the interfacial tension increased. And this was a property for a particular surfactant system. So we always have these surfactants in these oil-water systems and not for others. And the key thing here is that it gave us a handle to be able to sort droplets as a function of their interfacial tension and hence pH. And over here, I show you the simple device we created to be able to sort droplets as a function of pH. So what you have here is you have droplets, two population of droplets at two different pHs flow into the channel over here. Now let's focus first on the population of green droplets that are at higher pH. So higher pH, lower interfacial tension. They flow left to right and they encounter this rail over here where the channel is higher. Since it has space to expand, the droplets will expand, but the flow of oil will knock these droplets and they'll get reflattened, if you want, and flow over here. Now, the droplets at lower pH and hence higher interfacial tension will also encounter this rail and get closer to the spherical shape. However, because they have higher interfacial tension, the oil flow isn't sufficient to push them off this rail. So they follow the rail that's oriented 45 degrees relative to the direction of flow, and they move laterally in the channel, and they eventually the rail is tapered, so they eventually get knocked off over here. So you start off with two populations of droplets over here, two different pHs, and you've effectively separated them over here. So here's a video of this technology at work. We've added a bit of fluorescent to the droplets at low pH just to be able to differentiate the two different droplet populations. And as you see here, we can separate the droplets as a function of pH. So in a very simple way, it's a very simple technology in the sense that all we're doing is separating droplets as a function of pH. But it opens up a lot of interesting applications, which is going to be the focus of the rest of my talk. And we start off by using this technology to separate enzymes. So it turns out that many enzymes produce acidic or basic products. And here's one particular example where HCl is produced as a byproduct in this particular enzymatic reaction. And when we looked at the literature or databases, we found out that about 6% of enzyme produce an acidic or basic product. And a very conventional way of measuring enzyme activity for these particular enzymes is just using a colorimetric pH indicator to measure activity. Now, not only are these enzymes very common, they end up being quite important, particularly for bioremediation. So in this particular video, what we've done is we've taken an enzyme system, so in particular phenylacetate, added in esterases, and the product here is acetic acid. So in a vial, we started the reaction, put in a pH meter, and produced droplets at the same time on chip. And initially the pH was around 7, but as you produce more and more product, the pH slowly drifts lower. And the position of these droplets slowly increases on the rail. So initially, when it was at pH 7, the droplets falling near the bottom. And as the pH has drifted to around 6.8, we see now the droplets are going all the way to the top of this rail. Now what we can do is plot the displacement on the rail on the y-axis, and here is a function of time. And in blue and red is for two different enzyme concentrations. Now if we plot these two different enzyme concentrations as a function now of pH, we see that regardless of the enzyme concentration, the droplets will move up on the rail at the same pH or pH threshold. And this really shows that it's pH rather than another factor which is dominant for where the droplets will fall off the rail. Now this technology can be used to separate enzymes as a function of their activity. In this particular case, we use two different enzymes, two isoenzymes, esterase isoenzyme 4 and 5, that share lots of characteristics but have slightly different activity. Now, isoenzyme 5 has higher activity, produces more product, and therefore achieves a lower pH. So if we start both of these reactions at the same time, the droplets containing esterase isoenzyme 5 will achieve a lower pH and can be separated from the rest. Here we're showing just the separation of two different isoenzymes or different enzymes, but you could potentially see how this could be used to separate hundreds or potentially thousands of different enzymes to be able to do enzyme screening or potentially directed evolution. Now we started this work doing enzymes, but we quickly saw that maybe the most exciting application had to do with cellular sorting or cell sorting. So let me show you show our work on that. First, a little bit of background. So how do people typically sort cells? And the real workhorse of cell sorting is FACS, or fluorescent activated cell sorting. And here, generally what you do is you'll make the cells pass a laser beam one by one, and depending on whether the signal you get is below or above a threshold, you send it in two different directions. Now this technology relies often on different labels, and to be able to sort cells based on, let's say, the amount of antibody bound. So it's ultimately going to be limited by the sensitivity and availability of your probe molecules. And for this reason, people have come up with a lot of what are called label-free technologies, so they don't require a specific label. And here I mention a bunch of different technologies, particularly in the microfluidic field, where we're sorting cells without labels. And in these techniques, you're using physical properties to sort cells. So this might be their deformability, their size, their electrical conductivity. So you're sorting cells based on the physical properties. So it might also be good to figure out ways to sort cells based on what they're doing or their metabolic activity. Now, one more piece of background information that I want to introduce, and this is the idea of a Warburg effect, or what's also called aerobic glycolysis. Aerobic glycolysis. And this was discovered about 100 years ago by Otto Warburg. Now let's focus first on a normal cell. It takes up glucose and mostly undergoes respiration. Now the key thing is that cancer cells, their metabolism is reprogrammed. They take up huge amounts of glucose, and this is, by the way, the foundation of a PET scan. And not only do they take up huge amounts of glucose, rather than respiration or oxidative phosphorylation, they instead use glycolysis over here. So they secrete large amounts of lactate and a lot of protons. And this effect can be quite large. So if you put a pH meter in a tumor, rather than the pH being at around, let's say, 7.4, the pH can sometimes be below 7, or even in some cases below 6. So you create a very acidic environment. And this is one of the ways that cancer cells evade the immune system, is by creating such an acidic environment that immune cells can't function. All right, so we have these normal cells and the cancer cells. Well, if we put these two into droplets and wait a few minutes, the normal cell will stay near its initial pH. But due to the high glycolysis of the cancer cell, it will achieve a lower pH. Now, it should be noted that if we also have a normal cell or no cell at all, or a dead cell, it will also stay at its initial pH. Now, this gives us then a strategy to be able to sort these two systems. What we can do is we can take a cell, put it in a droplet, incubate it for a few minutes, then sort it based on the differences in interfacial tension as a function of pH. And in this case, we'll be sorting cells based on metabolism rather than physical properties. Here is our microfluidic device. Let me give you a brief walkthrough. We encapsulate cells over here into droplets. The droplets then go through a long serpentine channel. This allows time for the droplets with cells to achieve a different pH. And then we use a sorting rail, as we showed previously, that leads to two different chip outlets. It should be noted that we also use different surfactants in different parts of the chip. So the first thing we wanted to do was characterize the pH of individual droplets containing cells. To do this, we used a ratiometric fluorescent pH sensor, in particular pyridine, that shows a different excitation spectrum as a function of pH. And in this video, what I'm going to do is I'm going to scan from left to right, so in later times in our incubation channel. So the droplets are made. Initially, all the droplets show the same fluorescence. But as we go further down the incubation channel, so later in time after encapsulation, what we start noticing is some of the droplets are darker than the rest. Now, these are droplets that contain cells. The cells secrete protons, achieve a lower pH, and hence a lower droplet fluorescence. Now, we can look at the fluorescence in two different channels, violet and blue excitation. So what you have here is a bunch of droplets. Individual cells in those droplets are circled in green. Now, the droplets in the violet channel look brighter. The ones containing cells look brighter than the ones that are empty. And it's exactly the opposite in the blue channel. Now, what we can do is run droplets of known pH and create a calibration curve. And from this calibration curve, determine the pH of individual droplets. So each one of these points represents one individual droplet for empty droplets, and one containing 1, 2, or 3 cells. And what we see here is that droplets containing cells achieve a lower pH, and even lower if they contain multiple cells. And there's also a different range in different pH values, and this shows you that individual cells have different metabolic rates or glycolysis rates. So here's an experiment. So now that we know that the droplets have different pH values, we can use it to sort. And what we have here is a cell from a leukemia cell line. What's hard to see in this particular image is that we also have a bunch of white blood cells from a healthy individual or healthy donor. Now, most of the droplets are going towards this unselected exit. Now, the droplet containing the leukemia cell gets separated. So this shows us that we can separate cells from a cancer line from healthy cells based on differences in glycolysis. So I'll show this video one last time where you see this individual droplet gets separated. Now in that particular case, we separated, let's say, a cancer line from a healthy cell. What we also wanted to do was separate maybe on a finer line, be able to separate cells that show different levels of glycolysis. And the reason for this is that if you take, there's a lot of experimental evidence that the higher the metabolism of a cell, potentially the more dangerous or malignant that particular cell is. So can we separate cells that show different glycolysis rates? So here we have a video where individual cells have been labeled to render them fluorescents. And we're flowing droplets here from left to right. And what's hard to, what we're actually doing in these videos, we're actually constantly going between three different excitation colors. Blue, violet, and in this blue and violet will allow us to determine the pH of individual droplets in our system. So we focus in here on this particular droplet. Based on its violet and blue excitation, we can determine its pH. In this case, the pH is 6.64. Now we can also determine the pH of all the other droplets in our system, as you see here. Now the pH with the droplet with the lowest pH value will get separated from the rest. So this video shows us that we can separate droplets based on their pH value or glycolytic cellular glycolysis. Now we may want to separate droplets for different pHs, and to do this we have an external user-defined parameter, which is the entrainment flow, which is shown over here, up here, that allows us to set different thresholds for selection. So in this plot, we have the flow rate of this entrainment flow on the x-axis as a function of pH. Now the green droplets are droplets that get selected, and the red ones are unselected. And as we see, as we increase the flow rate, the pH threshold, or the threshold where we select droplets, decreases. So at low flow rates, we might grab or select droplets that have a pH, let's say, around 7.15, but as we increase the flow rate, we may be able to set a threshold at something like 6.5. So we can, using this parameter, we can choose which droplets we want to choose. So I mentioned before the Warburg effect. So the idea that cancer cells that want to divide quickly preferentially use glycolysis. It turns out that this phenomenon is not only limited to cancer cells, but a property that's shared with many fast-dividing cells. Here I'll give you the example of naive and activated T cells. So when T cells are activated, they also go through a Warburg type effect. So they ramp up, they start intaking more glucose and also ramp up glycolysis. What this means is that we can also put these inactivated naive T cell in droplets and then be able to separate the activated cells from their naive cells based on differences in pH. Now, a key thing to mention here is that the change in metabolism occurs minutes after the activation event. So normally, the surface markers that are indicative of activation usually take hours or even days to be displayed, while this change in glycolysis happens very quickly, which means we can separate activated T cells very soon after the activation event. So first we want to characterize the pH of droplets containing cells depending on the amount of time the cells were activated. So here we have naive cells that have not been activated and cells that have been activated for different amounts of time. And by the way, in all these experiments, the incubation time is always the same, about, I believe, 8 minutes for this particular experiments. And what we see here is that on average, activated cells achieve lower droplet pH, which seems to decrease with the time of activation. Now we started off with a proof of concept experiments. What we did is we activate some cells and did not other ones, so kept them as naive cells. We mix these two together, so these kind of shown by these green and red little dots. So we mix these two together in the vial, and we said, well, can we unmix these two populations? So here I show you this particular experiment. Now we again use fluorescent labels to differentiate between our naive and activated population. Here we see one naive and one activated cell. They have different pHs, and the activated cell will ride the rail upwards and get separated from the naive T cell. So this is often, these are plots that often show what our data looks like. So on the y-axis you have pH, and you have the two different populations. Here you have naive cells, and here you have cells that have been activated for only 15 minutes. They show a wide range of values, but you can see that on average, the cells that have been activated for 15 minutes achieve lower droplet pH. Now we can plot that same data, but as a function of unselected and selected data. So droplets have been selected and ones that have not. And in this case, the threshold was set around 7.2, or just slightly above. And all the selected droplets contain activated cells. So we completely excluded droplets that have not been activated in our selected population. Should be also noted that we don't grab all the activated cells. As you see here, in our unselected population, we also get some activated cells. But the key thing to note here is that activation isn't just a black and white effect. Cells can be more or less activated. And the more they're activated, the higher there's evidence that the higher the glycolysis rate. So in some sense, our technique is not just separating activated cells, it's separating the most activated cells, which is quite an interesting parameter. And there's also evidence that the higher the glycolysis rate, the cells that have higher glycolytic rate will lead to different differentiation outcomes. So we're really not only separating activated and naive cells, but we're separating a subpopulation of highly activated cells that could be interested both for biotechnology, but also for study. Now, I'd like to summarize our work. So we call this technology, by the way, SIFT, or sorting by interfacial tension. And we're able to separate enzymes and also cells. In particular, cells that are the same type. So cells that would otherwise share many of the same physical properties. The technique doesn't use any labels, excitation sources, active components, or detectors. So it's a completely passive technique. Because it's based on the physical properties of droplets, it's simple and robust. And it's also amenable to parallel sorting because it doesn't contain many components. Our selectivity is about 0.05 pH units, and the sensitivity can be modulated using our buffer capacity, so how much buffer we have in our device. Now our throughput is currently about 80 droplets per second, so substantially lower than some of these other sorting techniques, such as FACS, which might be 1,000 cells per second. So this is one parameter we're hoping to improve. So what have we used it to separate? And this is where I think this technique is quite special. It really has a lot of different uses. We've been able to show that we can sort high and low activity enzymes, droplets that contain or don't contain cells, live and dead cells, cancer, non-cancer, cells that have been treated different ways. I ended by showing you recent data where we separate activated and naive T cells, and also ones that have been tagged with light, which is something I haven't shown in this particular talk. So with that, I would really like to acknowledge the people who actually did this work. Here's a picture of maybe the students that have been involved in this project. I would really like to note out Dan and Xing that was involved in the early development of this work, followed by Claudia and Adriana that worked on this later on. So it's really a group effort, and I also mentioned some of our collaborators have been essential for pushing this work forward. And I would like to also end with the different financial support that made this work possible. Thanks a lot for your attention.